So in previous lectures, we talked about transcription, which is the process of making RNA from that DNA transcript. Once you've made that RNA, that mRNA, those instructions for the protein, and you've modified them to make them into transcripts and allowed them to leave the nucleus, you use them to put together amino acids. And as we've kind of talked about ad nauseum, proteins are these strings of amino acids all put together. And ribosomes are the things that actually put those amino acids together. They actually do the protein synthesis. They are a large conglomerate of enzymes and ribosomal RNA in two subunits. And here's a picture of that here. You can see here is the large subunit. Here's the small subunit here. And the mRNA feeds through this thing kind of like a tape. And as it's feeding through this ribosome, the ribosome is actually reading it and using it to say, okay, here's the protein that I need, or here's the amino acid I need next for this particular protein. And what it does is it has these tRNAs come in and continuously build this polypeptide, this protein up. Okay, and there are these EP and A sites. I'm not really as concerned with those. It's something that you should kind of know about. You should kind of have in the back of your head, but I'm not super worried about that. What I want you to know so far is that that transcript is made, it is modified, it exits the nucleus, it's going to go to a ribosome. The ribosome is going to read it, and it's going to use it to put together a series of amino acids. So, like I was just talking about, tRNA, or transfer RNA, is the molecule that brings the correct amino acid to that piece of, to that ribosome, to that mRNA that's specifying it. Okay, so what happens is those little things, those little tRNAs go, and they find the specific amino acid that they're looking for, according to that three-letter code presented by the mRNA. And those tRNAs, they know that three-letter code, they can read it, and they can say, okay, it's my turn, I got to go get this one particular amino acid. And so here's kind of like an image of what that might look like. You have your piece of mRNA here being sort of fed through this ribosome, and as the ribosome is reading each codon, these tRNAs can come in and attach here and add their amino acids to this growing peptide, to this growing protein. So as each amino acid is specified, those tRNAs are going to bring it in and add it to this growing chain here. And this is what your protein is going to look like. So as I've talked about a few different times, mRNA has these codons. It has these three-letter pieces of DNA that specify a particular amino acid. We even talked about the table and how to read those. And so this one is GCU here, and that's specified by this piece of mRNA. Okay, or you could think of it as GCA, and then this tRNA has the complementary strand, or the complementary piece of genetic information, and it's going to use that to go ahead and say, okay, I need to get an arginine now, so now I'm going to go get that particular piece of arginine. And they've actually done a pretty good job by reconstructing like three-dimensional shapes of this. They do kind of resemble these things with these loops in them. So mRNA binds to the small unit of the ribosome, the start codon, because remember in that genetic code that we looked at previously, there are things for start and stop. The start codon brings in the first tRNA with methionine and then binds to the large subunit. Okay, so tRNA is going to come in, okay, the ribosome says, okay, start, AUG, we've got our start codon here, and it's going to bring in that tRNA, which is going to bind with methionine, and it's going to start building that polypeptide chain. The tRNA that matches the next codon brings in the next amino acid, and it binds to the A site. So remember, you had those EPA binding sites, and the tRNA is going to bind to the A site, and the ribosome is going to catalyze the peptide bond between the first two amino acids at the A site. Then the ribosome is going to move along from the mRNA to the next codon. Okay, it's going to move the mRNA along, and the newly formed peptide is going to go from the A site to the P site. And that continues going until a stop codon is reached. And like I said, you don't necessarily need to worry about those binding sites. I just kind of want you to have an image in your head of how this works. If I asked you to draw a picture of translation, you should be able to draw a piece of mRNA in a ribosome, and you should be able to draw tRNAs grabbing amino acids and adding them to a growing peptide chain. Okay, once you get to a stop codon, the new tRNA comes into the A site and the whole complex falls apart, releasing the new protein, so that those ribosomal subunits are going to unattach from the mRNA. The tRNA is going to go away, and the protein is actually going to detach from the ribosome itself. Okay. 5 amino acids are added every second, and multiple ribosomes can move along a transcript simultaneously. I'll show you what that might look like. So you could just have another ribosome right here working on this same exact piece of mRNA, building that same protein. Okay, so this is a very, very, very fast and a quite energetically expensive process. So here is just a picture of it. You have your start codon, AUG come in here, which it actually codes for methionine. Proteins are all going to start with methionine, except for they're later on, a lot of the time, going to be modified to remove it. They might keep it, they might not, but they're going to start with methionine, okay? And you're going to see that this tRNA is moving along these various sites. It's starting at A, and then it's moving to P, and then it's moving to E as it's continuing to build this protein. Okay, and you can see that these amino acids are being chained together now. They're being stuck together, and they're actually forming that protein. Because remember, a protein is just a chain of amino acids. And you can see the growing polypeptide chain right here. So 5 amino acids are every second. Multiple ribosomes move along a transcript simultaneously. And this kind of brings us to this question. It can be used as a buzzword sometimes. What is a gene? What is a gene? What are your genes? Well, a gene is a segment of DNA that codes for a particular amino acid. A gene is a section of DNA that's going to specify, I said amino acid, it's going to specify a protein. So it's going to have a bunch, it's going to say, hey, I need to make this particular polypeptide, I need to make this particular protein with these exact 50 amino acids in this order. So simply stated, a gene is a segment of DNA that codes for a particular protein. So going back to the analogy of a recipe book, if you look up, chocolate cake in a recipe book, that would be kind of like the gene in this case. It's kind of a dumb example, but so a gene, piece of DNA that codes for a particular protein, just like a particular page of a recipe book is going to code for a particular recipe. And here's just a nice image of the process of translation happening. You can see there's this mRNA is being lined up, sort of strung through all of these ribosomes, and there's proteins actually growing off of them. This is a fantastic piece of electron microscopy. So in the next lecture, we're going to start talking about genetic regulation.